Okay, well, let's jump into this. Quick review of ASD Genetic Epidemiology, and the risk to siblings is about 20% and is about 20-fold higher than the population prevalence. Within monozygotic twins, the concordance is about 90%. That's a 90-fold increase. That jump compared to dizygotic fraternal twins or siblings is a lot of the initial evidence that led many molecular geneticists that weren't interested in autism into the field. And we continue to benefit from the fact that autism is seen by many major geneticists as a very important topic to study. To talk about genetic etiology groups, and this I would say is something that's a new emphasis in the last 10 years, is this top part that is de novo, meaning not inherited from either parent. These are almost by definition rare. They're predicted to happen in genetically lethal disorders, and that basically means disorder in which the chance of having children is reduced. And so if you look at particularly neuropsychiatric disorders, the two that have fewer offspring are schizophrenia and autism spectrum disorder, and particularly autism spectrum disorder. Certainly there are people with autism spectrum disorder who have children, but there are many others who do not. Now, I emphasize large here with the font, that these often have a large individual effect. So there are people in Mind Institute who are studying some of these. And their aggregate effect is, I'll show you later, is probably about 10%, although that number may go up. So again, for the individual, maybe very strong effect. For the group, is probably a minor effect overall. So then we go to inherited variants. These are variants that parents have that are transmitted to the children. There are rare ones, and I kind of made that a little bit smaller to be rare. They can have a large effect, especially when they're late onset. Now, that's not relevant to autism spectrum disorder, but it will explain to you why there are large effects when you think about Huntington's, because Huntington's does not affect whether you can have a child or not. Or they could be recessive, and this is a part of the, some cases of autism are related to recessive rare genetic changes. And recessive would mean you would have the same change in both copies of the two chromosomes. But what often happens, that you'll see that, Chris Walsh has found that when he studied some tribes that have smaller population size, there's more increased chance of relationship between people will usually lead to recessive cases. What's more common in larger populations are compound heterozygotes, which we look for, in which you have a variant that might be knocking out the gene on one chromosome and a different variant knocking it out on the other. That's a compound heterozygote that ends up being like a recessive. And then you have common inherited variants, and these are generally of small individual effect. And my example of the day is macro-D2, which has been in and out of GWAS studies. It is in the latest study. One of the interesting things about genetics of autism is to keep up, you have to search bioarchive.org as well as search PubMed. So the GWAS that I would refer you to is actually now in bioarchive, and in a format that I'm sure it will be changed by reviewers, but a very high quality. So again, small individual effect, and one of the things to point out, as I mentioned, macro-D2 has been significant and not significant over time. That tells you it's a small effect if we're only now saying it's significant at the 20,000 cases level. However, in aggregate, these small common effects are leading to the largest genetic effect. So accounting for about 0.4 to 0.6 of ASD risk, and but that's not from a single common variant, that's from hundreds or thousands of common variants. This genetic model of autism hasn't changed much in about 20 years. Probably should have published it instead of just having it in talks. And this is a model for autism genetics. And I make the case that most cases are multifactorial. Multifactorial is what was explained to me that disorders like autism would be like when I was in medical school in the 70s. And it turns out that was largely correct. Multifactorial implies that it's not all genetic influences. I'm emphasizing genetics because it's what I do. I'm very happy to know that people are studying the environmental elements here at the Mind Institute and everywhere else. And I could talk to you about how that would interact here, but let me focus on the genetics. Historically, this model was first presented in the 90s when the best model for autism was 2 to 5 variants contributing to autism. And that's why there's 5 overlapping circles there, and you would only have autism if you had all 5 risks together. Thinking of relatively common variants, they'd have to be rarer than we now know them to be. And that would actually be larger than this small area over here. This circle is meant to be a little smaller than that, again saying that these less complex cases that are largely de novo rare variants are this piece, but a lot of these. Now, we don't think this is 5 genes. Each one of these circles you would think of as hundreds or maybe even thousands of genetic variants. So that's the translation there. Less complex. Sometimes people call these monogenic. I don't think anything's that simple in autism genetics. I'll tell you later again why this is 10 to 20%. 10's my best guess right now. And examples are already mentioned: maternal chromosome 15q11, q13 duplication or triplication being studied here, 16p11.2 deletion or duplication, Fragile X extremely well studied here. And the first of these, I don't think anything predates Fragile X, although the association with Fragile X and autism predated finding that it was the FMR1 gene. CHD, loss of function SNV, and I'll be talking about many more of these single nucleotide variants and exactly what they are, and many other copy number variants in SNVs. Oh, and the one thing I want to say before I leave here is to make it even more accurate. It is not the case that this common risk does not have an impact here. That's why I call this less complex, because these common variants are probably impacting the severity or associated conditions of these rarer conditions. So let's step back and talk about, we're going to lead into CNVs, copy number variants, and I want to just talk about chromosomal disorders from an insertion and deletion perspective. So a chromosome segment is typically what you would find is an additional copy or deleted portion of a chromosome. The most common deletion syndromes, which typically range about 1 in 5,000 or less in the population, Williams syndrome, I've already mentioned 15q11, q13, both deletions and duplications, 16p11.2, 22q11, which I know is also studied here extensively, 22q13 deletion or Phelan-McDermid syndrome. There's autism risk in the above regions. In 7q11, it looks like duplication is probably a more common event than deletion, which leads to Williams. In 16p11.2, we have both deletion and duplication as a risk. 15q11, q13 duplication, and 22q11 deletion and 22q13 deletion, showing that some of the more common areas that deletions and insertions occur are having an impact in autism risk. So I have a preoccupation with 15q11, q13 region, and this is showing you 2 normal chromosome 15s, and I'm going to show you why I'm a stickler for calling it q11, q13. So a little chromosome nomenclature. The first is, this is the P arm, and this is the Q arm. Cytogenetics was invented in France, so think of P for petit, all right? So that helps you. Lejeune, who also found Down syndrome, was due to trisomy 21. Q is the bigger N. Now, right now you see all this 1.1 in the back. You probably can't see it, but there's a lot of 1s, and then there's a lot of 2s. When this was first done, the level of resolution is you just saw the 1s and you saw the 2s. There were really just 2 bands on the long arm. But then as the technique got higher resolution, then you had 1, 1, 1, 2, 1, 3, 1, 4, and 1, 5 within the 1 band. Okay, well that's okay. I see how you're going to go with this counting. Then they got logical as far as I'm concerned and started doing 1, 1.1 and 1, 1.2. Why they didn't just go 1.1, 1.2 in the first place, I don't know. Because I will be honest with you, you were told I learned genetics later than going to graduate school to learn it. And I kept wondering, if it starts with 11, where is 1 to 10? But if you think of it as 1, 1, it makes complete sense. So these are 2 normal chromosome 15s. In the case of Prader-Willi syndrome, this 1, 1 to 1, 3 region would be deleted, and it would be shortened. And Prader-Willi deletions, missing 15q11, q13 from the father, a range, but average mild intellectual disability, severe obesity, and OCD unrelated to the eating behavior. Actually, I think the same deletion on the chromosome that came from the mother will lead to Angelman syndrome, missing 15q11, q13 from the mother, severe or profound ID, often severe epilepsy, and ASD, although it's a little harder to tell at that level of intellectual function. So I'm partly talking to you about the difference between maternal and paternal because when we go into duplications, we also see different phenotypes. So the maternal duplication, this is what's called an interstitial duplication. You still have 46 chromosomes, but the chromosome coming from the mother has an extra piece in this q11, q13 region. Interstitial because it's within the same chromosome. In this case, which is actually more common, sometimes called inverted duplication 15, there's actually, if you count chromosomes, there's 47, with this chromosome having an extra piece of 15q11, q13. Actually, typically has 2 extra pieces of that region, and these are always from the mother. There hasn't been one seen from the father. That's partly because these chromosomal changes are more commonly coming from mothers and related to advanced maternal age, but not in all cases. So this is the duplication. We have the interstitials, which tend to be milder, and the inverted duplication 15, q11, q13, which tend to be more severe in terms of risk for epilepsy and intellectual disability. This is the first case that had me interested in this area, which is, this is G-banding, which is a terrible way to find these interstitial insertions. She doesn't have an interstitial insertion. It's easy to use G-banding to find out you have an extra piece of a chromosome. This you will never see except in this talk probably. This is C-banding and simply shows you that from a physical perspective there's 2 centromeres. This woman, who I still see many years later, presents with severe intellectual disability, seizures, although fairly easy to control, repetitive behaviors, anxiety about lack of order, self-injury, mood lability, and hyperactivity. She had a very nice response to carbamazepine and an SSRI over the course of about 20 years now. This is about 0.3 to 1% of ASD, and this is the best way to learn a lot more, particularly to pick up the mild dysmorphology of this syndrome, is to go to the dupe15q.org family website, because families can put up pictures of their kids a lot easier than we can show them. And there's a lot of useful information there. This was presented to me in the early 90s as she has this extra chromosome and it doesn't matter. And the father, who is very astute, said, "How do they know?" So I'm going to show you now an interstitial. This is metaphase fish. Basically, what this is showing you is that there are probes that are showing you 2 instead of 1 copies on one of the 2 chromosome 15s there. This was a family with that interstitial duplication that we reported about 20 years ago. Basically, somewhat by chance, as I was telling the postdocs and students today, had 3 alleles at this one location and several other locations in this 15q11, q13 region. And this is a duplication. Now, hopefully, I can walk you through the fact that you have the mother's mother and the mother's father. She should get a chromosome in this region marked either by this or by this. And you can see simply that she gets 1 chromosome from 1 haplotype, a set of markers from her mother. So she's only getting 1 copy from her mother, but instead of just getting the 139 or 147 from her father, she gets both of them. So she has a duplication that came from her father, and she's fine. But now the exact same duplication has a 50% chance of being transmitted, and when it's transmitted from the mother, not from the father, both of the children with the duplication have autism spectrum disorder. So that's showing in a similar way to the Prader-Willi-Angelman syndrome that there are 2 different phenotypes for the deletions depending on the parent of origin, and there are 2 different phenotypes for the maternal versus paternal duplication. Before we published this family, the discussion was that a duplication coming from the father would not be compatible with life, and I assure you the mom is very, very alive and lively. This is the duplications coming from the Simon Simplex collection. You see that there are roughly 2 classes of them. Some are a little smaller than others. That doesn't seem to matter, so the real emphasis seems to be in this region, and work here at the Mind Institute is emphasizing UB3A, which makes sense because UB3A is the gene in the region strongly expressed from the mother as opposed from the father. So now I want to go back to the larger discussion of copy number variants and single nucleotide variants. And so a copy number variant is an insertion or deletion, and it's a missing or extra paragraph or page of a book, and they're typically relevant when they delete or add genes. A single nucleotide variant, in a missense one, changes the amino acid of a protein. A loss-of-function single nucleotide variant is a change in the nucleotide which goes from an amino acid to, say, stop making the protein. So it'll lead to a truncated protein, which is commonly not functioning. So I give you the example, supercalifragilisticexpialidocious. And yes, it is interesting that Word picks up misspelling that, but will not suggest how to fix it. So this is the expected word. So you could think of this as amino acids in a short protein. And a missense de novo mutation in one of the child's 2 copies is this F changing to an X. And de novo nonsense, loss of function, was just super cali because it said instead of adding the F, it didn't say add the wrong amino acid and keep going. It said instead of adding the F, it said stop. Okay? So the code was changed by that single change. Now, one of the points I want to make is that we all have, on average, one of these happens to us. All of us. So if I look around the room, if one person has 2, then somebody else randomly has 0. It's very interesting because in the autism exome studies, that was something that had been predicted, but was actually observed as sort of a human genetic mutation rate. The difference between that mutation that on average we all have one of is, does it hit in a location that leads to loss of function, which are going to be the strongest effects? Does it have an effect on this readout? This might disrupt the protein as much as shortening the protein. Does it actually lead to a change in the genetic code that doesn't change the protein at all? These are called synonymous. They're a change. They don't have an impact. Obviously, the ones that, well, I don't know, obviously, the ones that don't affect the protein are not leading to risk for autism and much of anything else. There are some exceptions, but if you look at 1,000 people, that's the case. These are the ones that have the strongest effect. And I'll drill down a little bit to that in autism, more specifically. So we're going back to CNVs a little bit. These are 16p11.2 microdeletion in autism. This was just, there were actually 3 papers that came out about the same time, and that was 2007. And these are basically deletions or duplications below the level of resolution of the microscope. So those 15q11, q13, especially deletions, are right at the threshold of whether you can pick it up under the microscope. When you get to the 500,000 base pair level, you need array-based methods. This is that region zoomed in, and as soon as, for example, our group and others found that deletion, the next step was to look at the genes in the region and see if they had mutations that might, in and of themselves, lead to the syndrome. And the answer now, 10 years later, is you can't point to a single gene in this region that has any SNVs or specific mutations in a gene to suggest that it's one of those genes solely contributing to the phenotype. So the CNVs that contribute to autism spectrum disorder tend to have multiple genes affected, and in most cases, it's probably not any single gene sufficient to cause the phenotype, although, yeah, that's probably generally the case. I'll talk more about that later. So this is a review from about 10 years ago, and it turns out in the array space there are other duplications and deletions, but much of what we knew about 9 years or 10 years ago. And you'll see maternal duplication 15q11, q13 on the left there. You'll see paternal dup 15, q11, q13 on the right. What you see on the left is a CNV is enough by itself to put one over the threshold for ASD. The darker gray in this case is other genetic liability and non-genetic factors. And I'd like you to think of that not just as additive, but also potentially subtracting. So it might pull someone down below a threshold as well as push them over. So the ones on the far left seem to be enough to push you over the threshold by themselves. As you move across from the right-hand side, you're going to need other genetic liability and non-genetic factors. The other genetic liability might be polygenic risk, those common variants of many of them, and non-genetic factors are many things contributing to risk for autism. Okay, we're going to shift to whole exome sequencing. And that shifts us largely to more discussion of de novo single nucleotide variants. With microarrays, we could find copy number variants. It turns out we find some additional information from whole exome and whole genome sequencing in terms of copy number variants. But the increased resolution of whole exome and whole genome doesn't make as much of a contribution as you might think, because it's the larger copy number variants that matter. In other words, thinking about losing 3 pages, it's taking out 3 genes instead of just missing a sentence that might be in between the coding region of a gene. What we know is that there's a higher burden for loss of function de novo SNVs and a smaller enrichment of de novo missense SNVs in brain-expressed genes intolerant of missense mutation. I think I need to translate. So one of the things it says is that it doesn't matter if you have a change that doesn't change the protein. If you have the change like the supercali that I showed you, where you now are cutting the protein, you're basically eliminating the function of one one copy. Now there's usually still a normal copy because these things, it's not lightning hitting the same gene twice in this case. And this is where the intolerant omniscience, actually this is also a case, this point about brain expressed genes, it only makes sense that the gene's not expressed in the brain, and there may be some exceptions, but as a large story, genes expressed in the brain that now have a loss of function are more likely to lead to ASD. Intolerant of mutation, I mentioned intolerant of missense mutation, but it applies both to loss of function mutation and missense mutation. Some genes can simply tolerate missing one copy being functional. They don't need the proper dose. There are probably some that are tolerant to even too much in the case of a copy number variant. The same thing is true of missense variation, and there's this very useful browser, if you're at all interested in this area, that will show you for a particular gene whether that gene is tolerant for loss of function mutation, and you could also look up a separate statistic for missense variation. And this is based on about 60,000 samples, largely controls, at least for ASD. Turns out in schizophrenia, you need more access to the data because there's some schizophrenia samples in there. But basically, they're showing you here which genes will tolerate the kind of variation that you'd be surprised. And I have to step back a little bit and talk about the personal experience of seeing whole exome data back the first time. I had always been taught that we all have blemishes, we all have variants that put us at risk for many disorders. I think the quote is about 7 potentially lethal disorders if they're on both copies. And so as you get these data back, the first thing that strikes you when you get whole exome data, and it's worse than whole genome data, is you just see all of that. And you will see a lot of genes that are tolerant of basically being knocked out in one copy, not necessarily 2. So these are 65 genes implicated in ASD, and I add FMR1 because FMR1 was excluded because it was thought to already be known. So when you start seeing some of these lists, you need to know what was excluded. 15q11, q13, and some other chromosomal disorders have also been excluded from some samples. So some of the larger samples may de-emphasize some of the things we already knew. And that means that almost everything in this list of 65 SNVs associated is something new. There are a few exceptions like SHANK3 that have been implicated from Phelan-McDermid syndrome. I'm not going to go through all 65. This number is in flux. It's probably already over 65, but we don't have a major paper showing what that number is. The estimates are going to go up to the hundreds, and you have to put that in the context of how many brain-expressed genes there are. The ones that I've underlined, GRIN2B is otherwise known as the NMDA2B receptor, which is an interesting target. It's something people have been trying to target pharmacologically already. GABA beta 3 is also a more traditional receptor target, and SLC6A1 at the bottom here is the GABA transporter, which is also a more logical target. That doesn't mean the others aren't important. It's just given some mechanisms of drug treatment, just focusing on the ones that might fall into the usual treatments. I know that people here are studying ARID1B and CHD8, right? So there's ARID1B, CHD8, these are alphabetical. As it turns out, CHD8, ARID1B, DYRK1A are some of the more common ones. SCN2A is a very interesting one because of its association with epilepsy as well. I'm mentioning SYNGAP1 because a patient that I followed for about 20 years, once we got his data back and did clinical confirmation, found that he had a SYNGAP1 mutation, which in terms of complexity is interesting because his mother has a brother with autism who you would have guessed there might have been an X-linked relationship. It obviously been screened for Fragile X, but as it turns out, whatever the brother has isn't what the son has. Whatever the mother's brother has isn't what my patient has. Only showing that within a family you can have heterogeneity and complexity of mechanism. GABA beta 3, the GABA A receptor beta 3 subunit, is the only gene implicated as a significant de novo SNV that occurs in a genome significant CNV, the Dup15q11, q13, but even there, we actually would think UBE3A is thought to play a more important role because of its maternal expression. So there's a region where we think there are probably two things going on. This finding of it as a de novo SNV could be independent of its role within the CNV. But I do think that the implication of only finding, basically what we're saying is that's the only one, meaning all the other CNV regions do not have a de novo SNV. And so the implication is that most CNVs represent a contribution of several genes deleted or inserted, which is consistent for a higher risk for de novo CNVs containing more genes. So remember I was throwing around that term, or that number 10% related to de novo rare events, and some people thinking it's higher. So I'm going to walk you through the 10%. Exonic de novo CNVs, 5%. Loss of function de novo SNVs, 10%. 3% for rare autosomal complete loss of function, the recessives, and they could actually be homozygous or complex heterozygotes. And then, of course, here we're aware of Fragile X, so you get rare X-linked homozygous loss of function, and about 1.7%, which are all male. That's 20%. So some people go, oh, we found 20% of cases of autism explained by this. And your logical question is, well, what were the rates in the sibling controls in something like the Simon simplex collection? And the answer is 10% of controls had that. So what I didn't really show you is the relative risk ratio, which is highest for exonic de novo CNVs, of about 5 to 1. So, for example, 5% of the cases had exonic de novo CNVs, but so did 1% of the sibling controls that did not have autism. So the net increase is 10%. Does that mean that in my clinic, if I do 100 arrays and 100 whole exome screens, or perhaps think that the whole exome or the complete genome is sufficient without that, am I going to get 10%? Well, no, I'm probably only get about 3 to 5% that I can interpret, because that 10%, we don't know exactly which 10% matters in a sense. Just to give you some sense of how dynamic this is on the sort of clinical interpretation side, the Simon simplex collection, which started 10 years ago, and I think we finished about 5 years ago, the clinical committee that looks at the research findings and decides whether we have something that we should go back to families with meets yearly, and every year there are genes that move from, we really don't have enough to go back to families, to we now know enough about that gene that this is something that people would want to know, and of course they consent it ahead of time to wanting to know about such things. And you may have questions later about terms related to how do you go from basically research findings to clinical findings. This is more of the same story. One of the studies from Sweden basically showing from a genetic perspective, again, about half common inherited effects. Those are not diagnosable things. 3% rare inherited, about 4% non-additive, and 3% de novo, but again, only about 3% of that. Here you're coming up with that same 10% number. Only about 3% of those are where you know the specific gene's impact. So the point of all of this is to try to develop new treatments. At least it's the point for me. Actually, I'm a clinical pharmacologist who got stuck in genetics for a while. So one of the big questions, we have many rare CNVs and SNVs. A few of them recur, things like CHD8, DYRK1A. One of those slides showed that whether this had been found in 2 people, 3 people, 5 people. There is a clustering among a relatively small number of pathways with synaptic plasticity a common theme, and a few final common pathways to mechanism may exist. So that's a somewhat hopeful statement. Although I suppose if we have to just slug it out gene by gene, then we just have to get on with that. This was from the early CNV studies, and it was very interesting to look at the genes that were being hit in actually both, all three, autism, intellectual disability, and schizophrenia. And it's interesting along this pathway from orexin in the presynaptic terminal to neuroligin in the postsynaptic terminal in these PSD95 associated proteins, that it's very interesting because the ones that were not hit were later found to be hit. So you have this whole chain, and it doesn't mean that people with autism spectrum disorder had all of them hit. It was sufficient if any along the chain was impacted. This is from the Simons study. This is actually putting the Simons study together with the ASC collection to date, the ERA studies, and this, I do need to make a comment here. The big change in autism genetics is that in 1990, individual labs thought they could solve the problem. And somewhere along the line, people formed consortia. And they were actually many investigators. And they thought the consortia could figure this out. And what happened is the consortia got together with the other consortia. And this has been a very positive thing in the field. And basically you have large numbers of people working together, very large numbers of families contributing. But I'm a little, I must say, I would say, I see Sally Rogers nodding. There's something very interesting and very positive about the development of the field over 25, 30 years, where it became much less the individual thinking they could solve it to a much more social process. So on the upper left, well, I'm going to focus on the bottom right. The bottom right has genes that many of you will recognize. In orexin 1 I had mentioned before, GRIN2B, or the NMDA2B receptor, many synaptic genes, GABA beta 3 is in there. We're not surprised to find synaptic genes in autism. The upper left has been more surprising. The upper left is really more about gene regulation and chromatin regulation. And I think that, you know, yeah, sure, it's been around for a while, so of course, yeah, we knew that was the case. But I don't think anybody predicted that that would be such a big piece of this. Oh, and what do I think is going on with the chromatin regulation? You know, this is just wild speculation, but, you know, what the heck. And a lot of this work has been done here and with collaborators from around here on the basic neurobiology. We know that there's a Goldilocks approach to just the right amount of regulation of turning genes on and off. If it's too fast, there's a problem. If there's too much, there's a problem. And if you disrupt these genes related to chromatin regulation, you're probably disrupting this very fine-tuned process related to experience-dependent plasticity of synapses. The fascinating thing is that some of these genes aren't causing more problems for the overall body and other tissues in the brain. I think it's just that learning is one of the most sensitive to dose. Many of those genes, if they were recessively knocked out and not just reduced in dosage, probably would not be compatible with life. And the other thing that's obvious here is that these genes, when I say chromatin regulating, I could also say very related to epigenetics, which I know people here are studying and are very interested in. Okay, etiology to neuropathophysiology. Maybe I should start this section with, now I'm going to speculate some. So most are related to reduced function, heterozygous knockdown. This makes them amenable to RNA knockdown studies, which many of you are doing, transgenics, which many of you are doing. I mentioned CHD8, and now I know people here have their own opinions on this, but it is the most common recurrent de novo mutation in ASD. And from a mechanistic, not a behavioral perspective, I was very interested in the approach that this one paper took looking at the pattern of gene expression changes being consistent with overall delayed brain maturation pattern. I just think it's good that we are prepared to find global effects as well as very specific effects. And I like the idea of a potential overall delayed brain maturation pattern for perhaps one or a set of these genes that are implicated. So let me just talk about a couple more. I actually have encouraged some students and postdocs at lunch and others that this is one you might want to look at. It's very interesting. I haven't studied it. I know about this because I hang out with diabetes genetics researchers that actually they're more friends than anything else. Excitation, inhibition, and balance. Huda Zogby talked about quite a long time ago. Others talk about. And I'm just going to give you a really quick example to look at. And this is KCNJ11 IDEND. It stands for diabetes, epilepsy, and neurodevelopmental. And it's a fascinating mutation because basically what happens is instead of ATP shutting this potassium channel down, the potassium channel is stuck open, and there's a hyperpolarized membrane leading to decreased secretion. This was initially discovered as a mechanism and is best worked out in the pancreatic beta cell that secretes insulin. And you see the diabetes piece of this. It leads to persistent neonatal diabetes, also ASD, ADHD, ID, and epilepsy, particularly in one of the more common missense forms. And there's a correlation of the KATP channel dysfunction. The correction of it by sulfonylurea restores calcium-modulated secretion of insulin. So basically, sulfonylureas, which are usually used in type 2 diabetes, will shut this channel, and instead of a hyperpolarized neuron, hyperpolarized beta cell, I get ahead of myself, it now has a normal minus 70 millivolt potential, similar to neurons, and the usual calcium modulated secretion of insulin can occur, and you have these patients that have been on insulin who can be treated with an oral agent. And that's one of the reasons I hang out with this group is it's pretty interesting to have a genetic condition that's so easily modifiable. A lot of people should know this just because it's a very positive genetic study. Now, the other piece of this is the ASD, ADHD, ID, and epilepsy, particularly with the more common missense variant. It's about an IQ in the 50s. I go to the group and offer, they kind of bring me in because these are the kind of problems that these kids have. And I feel like I'm in an autism group. I actually feel like I'm at a Dupe 15 conference. And initially I thought, well, wait a second, this beta cell actually looks a lot like a RAF8 neuron, except it's in the pancreas, if you look at the developmental program for it. So I think, oh, maybe that's 5-HT, and the hyperactivity, maybe that's dopamine, let's look under the street light. And then one of my colleagues pointed out, this is in all neurons. So the model I want to get back to with the details of this, people should study this better. I think I'm surprised more hasn't been done on the modeling, for example, is that this is really an inhibition imbalance. There's too much inhibition. And what's fascinating is that the different levels of severity of the channel problem, you see different, what appear to be circuit problems, but they're really circuit problems because global inhibition at different levels is going to have different impacts on different circuits. At the worst level, inhibition of the brain leads to epilepsy and death because you've lost, basically you get a disinhibition. You've inhibited the inhibitory neurons. But at other levels, you see other what appear to be very specific circuit deficits, but the initial problem is this overall inhibition. So anyway, approaches to studying EI, balance, transcriptional regulation across disorders is a challenge for many neurobiologists, including those at the Mind Institute. But, would you do that, or would you rather just knock it out gene by gene, mechanism by mechanism? I tend to be a little old school when it comes to that kind of research. But I'll leave that to you guys to make those decisions. This is overlap of ASD genes with ID, epilepsy, and other medical disorder genes. And just to point out that they do overlap, the epilepsy, you'll see some that are overlapping between schizophrenia, intellectual disability, autism, metabolic disorders. CHD is congenital heart disease. So some of these, what it tells us, and not surprisingly based on what we know about genetic syndromes, is some of them are going to be associated with medical comorbidity and some less likely. I'm going to jump from that to some ethical implications. I really have emphasized the complexity of genetic effects. I think it's very important as people start to talk about what we know that we're very honest about the complexity. I've left out some things like in 16p11.2. If you knew in utero that somebody had 16p11.2, the only thing you'd be certain about is that they would have problems with severe obesity. They'd be at risk for neurodevelopmental disorders, but you couldn't predict that they definitely would have them, and they might be mild language problems. Subthreshold ASD traits are often adaptive for individuals without ASD, for example, capacity for intense focus, as well as may be adaptive for those with ASD, and of course part of our work is helping people in that way. Third is probably the most important implication from the polygenic risk. If you attempt to eliminate common variants for ASD, you'd have to eliminate the species because they are abundant enough that we all have many of them. We may not all have de novo loss of function variants contributing to ASD, but we all got plenty of polygenic risk variants. And there's a reason that they're there, meaning they're conveying some advantage in some context, whether it's current or historical. Beneficial effects of risk variants. Anxiety, another way to put that is avoids excessive risk. It's a balance, right? Restricted interest, my favorite. Ability to focus intensely. Every researcher can relate to this one. Okay, so if you don't have enough of those ASD-related genes, probably shouldn't do research. Another favorite is social impairment. And I have this one. Inability to lie well. Okay, so some of my parents say, oh, my kid's lying all the time. I say, yeah, and you catch him every time. You know? So, and actually, I'd like to hang out with more people that have this quote-unquote impairment, frankly. Context is essential. Gene-gene interactions, gene-environment interactions, and you know that quickly gets to be even more complicated. So a few more implications. Genetic etiology doesn't, some of these are like implications I don't want people to be still having in their heads. I don't think of them, but I know that people have these confusions about genetics. Genetic etiology does not reduce the need for habilitation, education, non-genetic treatments. And the best example is PKU, dietary intervention, prevention. The idea is to empower those with ASD and their families. Common risk variants for ASD are shared partly by all. And as far as I'm concerned, the point of genetics is to help choose available treatments, which I don't think we haven't been, it's not working as much as I would have liked it to by now. I do have this one, and it's a caveat. In my office, carbamazepine is often affected in the mood instability in duplication 15q11, q13. Particularly in the ASD and mild to upper moderate ID. That's something we're writing up. And it also leads to an interesting question of how would I actually conduct a trial to demonstrate that in a more convincing way than a case series. And then I really think the emphasis is to develop new treatments through understanding of pathophysiology. So thanks to everyone. Funders, many, NIH, Shaw Foundation, Autism Speaks, Simons Foundation, the American Academy for Child and Adolescent Psychiatry, and others. Everyone who advocates for services and research for those with ASD, everyone who cares for themselves or others with ASD, people with autism and their families who have participated in studies, Mind Institute for the invitation, and all the work that you do, and all of you for your attention. And I'll just finish with a picture of my brother And you notice he's enjoying running here, so you heard before about me enjoying running. And I can tell you that I thought of him, who had already passed away, when I finished that first marathon, and yes, I did cry. So thank you very much.
I was wondering if you knew what kind of impact an increase in potassium had on the deficit, the global deficit of potassium in the brain.
Oh, in the case of that, casein J11, it's specifically that channel. It's not an overall change, it's really at the membrane. Because of the opening of the channel, the opening of the potassium channel leads to a more negative membrane potential and less likelihood of secretion of insulin. The overall potassium level would not change, just the concentration gradient across the membrane.
Do you do research in neurofibromatosis and similar disease and how they're connected to autism?
That's a very good question. It actually reminds me of once writing a review article about genetics and autism, and I was told that there was not a connection between neurofibromatosis and autism. One of our Simon simplex collection families came back with a neurofibromatosis 1 mutation, and there is a relationship. It's an interesting one because neurofibromatosis would certainly fall into the range of one where there's a lot of variation in outcome related to autism spectrum disorder. But of the many learning problems related to it, autism spectrum disorder does seem to be one of the risks.
Thank you so much for this great presentation. There is a slide actually that it's from the paper Cook and Scherer, Nature 2008.
Yeah.
I'm not sure if it will show anyways. So there are different, as I remember, okay, good. I think it's, yeah. So, it's coming. Okay, yes, this one. So I just wanted to know what makes the first, let's say the first group of CNVs contributing solely, let's say, to the phenotypes of ASD? Is it because of when mapping these CNVs into genes, is it because of the gene function, let's say synaptic genes? I just wanted to know what's the difference.
I'm going to have to say I don't know. Okay. I think it's going to come down to which genes are in there and what their impact is. I do think is a fascinating, I'm going to take that question a step further. Some of these syndromes are more constrained for their phenotypic presentation. And exactly why that's the case probably does have to do with the specifics of the genes. So a lot of the emphasis is on overall burden for loss of function, or here's some genes contributing to autism. Some are associated with large head size. You know, some are associated with cancer. It's a good opportunity for me to follow up on why I had P10 bolded. I didn't mention. P10 is one of the ones we really need to think about with large head size, and particularly, you know, especially in the larger head size, let's say over 3 standard deviations, and you've got an expert in Professor Emerald to tell you exactly how to analyze head size. And one of the reasons to think about P10 is P10, increased head size, increased risk for cancer. And so as we think about the disorders we find, some of them are going to have implications for medical follow-up more than others. And again, that's going to have to do with where they're expressed, when they're expressed. And as far as the CNV risk, I just think it has to do with which genes are in there and what contribution they're having. So that's a whole nother story. And one of the members of these autism consortia that I really point people to a lot is Catalina Bettencourt. She comes much more from the medical genetics side, and you know, I'm much more, okay, we have all these numbers and statistics and Catalina's going, but that, you know, that KCNJ11 is an ID gene. It is an ID-ASD gene. It's on lists that she compiles that don't all come from these large statistical studies in a sense. And she'll know, some people can do that encyclopedic thing, I can't. It's pretty fascinating. And they should go on to clinical genetics. What I wanted to follow up, so we actually have a case, and I hate to say I can't remember the gene exact. Sometimes these, if you think these letters and numbers are bad, you're right, okay? I mean, I have a patient with this, and I can't remember it right now, and it's not just anxiety, but it is a lot. And so this patient, it turns out, has, especially for the clinicians in the room, has a verbal IQ of 130. He has mild ASD. He has visual spatial function in the 60s. It turns out his gene is highly expressed in the visual system. That's a very specific phenotype as opposed to something like 16p11.2, which may have a broad phenotypic expression. And that, again, with this NR2F1, I don't count on it. I'm going to look it up. Anyway, we're actually writing the case up. In that case, it has a very localized and strong expression. But that, I must say, it's interesting because it's such an interesting correlation with phenotype.
Again, thank you for being here this evening. And a heartfelt mom with a child with autism. So my question to you, it really has to do with genetics. Because I'm a social mom who started a social group, and as mothers have the same question. What about our kids? And why I ask that is because my research started at the campfire at the family reunion. And I found that 3 of my cousins and myself all have an autoimmune condition. And I recently found out we have a risk of having autistic children, but we all have autoimmune anemia, which is called spherocytosis. Okay, that's great. But we all have an autistic child, and we all think they're wonderful. But what about my daughter, who is typical? She says, Mom, you know, I know I also have spherocytosis. Do I or don't I have children? I said, well, you know what, I'm not God. But you have to really think about that, and when it comes to that time, maybe we'll know that there's a blood test you can take, you and your spouse. So what can you tell my kid and all the other kids who have the same question?
It's a very, very tough question. So I think let me try to unwrap it, and you may have to get me back on task because I can think of about 3 different points here. The first is the obvious personal question of choosing to have children and risk for a whole multitude of things. And that's really not what you're asking me about, but that's an important thing. I mean, certainly as a parent I can tell you when we decide to be parents, we don't, our kids aren't always healthy. And we have to be prepared for that and love them whoever they are. In terms of the risk question, there's a broader question in that, which is what's the risk? We know it's a high risk if the same parents have another child. That recurrence risk to siblings we know is quite high. The number drops considerably now I'm not talking about the spherocytosis and the autoimmune disorders. I'm talking about the general question of what in a sense is the risk to the, what's the risk of the niece and the nephew, which is basically what you're asking about, right? So it's the sibling's child that's going to be a niece or a nephew. It turns out the best data we have for that is it drops off probably not to the 1% in the population approximately, but it may drop down to as far as 2%. right? And then you got to think about that in relationship to the risk of you got no family history, you ended up with schizophrenia, and a number of other things. So, you know, now, I agree with you on the question of the spherocytosis. At the time it comes, that's a really good time to go and get very good genetic counseling, to understand, maybe some testing involved. Tends to be better with your daughter, not with her brother, if I read it right in terms of relationship. And we're learning a lot more as we go along. And then the thing is, if there's a family history, that becomes a completely different story. I showed you the one case, one of the interesting parts about the mother who had the paternally inherited, but then she passes it on to the children, is an example of how you can have a kind of reduced penetrance, which simply means you have the genotype, but you're not affected. She didn't, in a sense, have the same genotype because it's a duplication, but from the father, not from the mother. But, you know, you don't know just because she has a spherocytosis and not autism that isn't related to the risk. The other piece that we're studying, meaning the field is studying, is there a female protective factor? What is the nature of, what's driving fewer females to have autism than males? So that would be another reason why. Anyway, the very tough questions is definitely a question when the time comes. Very personal questions, good. And genetic counseling isn't just about genetics. It's part of the point. Oh, I don't know who's coordinating.
Hi. I'm actually a teacher. I'm not a researcher, but I do have a child with autism. And some time back, I guess I read about the how they were studying. There was a study in Wired about the parents that were working in industries where they didn't have contact, and they weren't real social, but they were very smart, and they had a high prevalence of autistic kids of different levels. Is there any other studies that have looked into that and seen if that was maybe just a fluke, or if it really is a prevalence there? I mean, maybe they were undiagnosed as being autistic, the parents?
I mean, I don't know the specific study or that it was a study other than a collection of stories. It's one that we've told in various university communities from time to time as well. And it fits the whole other line of, in the polygenic risk, and again, it's a little bit related to what I was showing, is you can have traits that are related. There's an interesting thing that I'm going to go ahead and throw out there because we're discussing it in the genetics field, which is that we're not surprised when we do the polygenic risk scores and the ones in autism correlate with schizophrenia. There's some overlapping social difficulties. We are surprised at the correlation with the best proxy we have. Well, it's called educational attainment, and it's partly because some of the longitudinal studies that have looked at it haven't had IQ, but they've had how far people have gone in school, which is a little bit of a proxy for that. And what they found is something that the first time we saw it, we're like, well, that's interesting, I'll throw that away, I don't need to worry about it. The second time we saw it, it's like, well, now we got to struggle with the fact that the autism polygenic risk score is correlated very paradoxically with higher, not lower, educational attainment. We've been struggling with this, like people that frankly can analyze genes better than I can, and we're not sure what could be going on there. But the point is that these are a lot of twists and turns, and it really does get back to the fact that there are advantages to some of the risk for autism. And frankly, if there wasn't, these variants wouldn't be around. That's sort of a corollary.
The pancreatic example is really interesting. And I want to know whether or not you know of expression differences in the autonomic nervous system. We tend to be, synapse in the brain centric, but we have all this autonomic misregulation that often accompanies states of emotional dysregulation.
Yeah.
And may be in a feedback loop with processing the world. So do you know anything about differential expression relating to ANS versus central in the brain neurons? That's one question. The other is, do you have any sense of this chromatin regulatory epigenetic piece and a non-pharmacological, non-genetic environment? So what would be your ideal conception of the kind of the enriched environment for individuals with autism that pushes that advantage traits that you've just been talking about in that direction and minimizes the challenges?
So for the last one, which I can remember, I remember the first one, but I'll tackle you have experts like Professor Rogers to help you with intervention. I can't see how I'm going to tailor the interventions based on knowing about chromatin regulation, but there are people here that may sort that out. I throw these out. There's so many I would say that the best thing that we've done in genetics is given a lot of people things to focus on. Of all the genes in the world, which one are you going to focus on? Okay, look at chromatin regulation the way you're talking about. This autonomic regulation point is a very good one. And it also reminds me that it might be more amenable to study than, there's a group at Southwestern studying visual system. I go, why do you want to study visual system in autism? But it's just so well mapped out. It makes sense if you're talking about potentially larger scale issues than, let's say, auditory cortex. Not that that might not be the more central thing I would think of first. And your point of something's accessible in the autonomic area, and there certainly is interesting work in the autonomic field in autism. So that's a really good point about something that might be more accessible, and something to think about even in the many models that people are working on as well.
So, Ed, you made a very good point about autism being multifactorial and polygenic. So is anybody...
More than polygenic.
Yes. So presumably that means that a given person has 2 or 3 or 4 or 5 variants or mutations. Have you or somebody else in the consortium tried to find within a given individual which people have which sets of which mutations?
So, we probably have more like hundreds. Oh, I don't know if you're talking about the rare variants that we have or the...
All of the above.
I'm really thinking of the polygenic side, because in a sense we all have a de novo rare variant, but most of us don't have one that matters. And I want to make another point about those de novo variants, and is that those de novo variants are somewhere along the line, those de novo variants are why we can communicate in the way that we are. Okay, so we sometimes focus on this de novo variant has led to this difficulty, but those de novo variants, mutations, sometimes leads to advantage. And I'll give you another example of that. We all know, we may not all know, ApoE4 is really the only really strong common variant in complex disease. And it's a risk for Alzheimer. Well, as it turns out, ApoE4 was the original protein. And the E2 and the E3 forms were mutations that are protective. So we tend to think too much about mutations as problematic, but the things that drive things forward and protect us are often adaptive mutations. And that's also important to put in the context of if one of us meaning in the population at large, has a mutation and confers challenges, maybe requires more care by others, we have to recognize if you get rid of that process, you get rid of the adaptive process of mutation.
The UC Davis Mind Institute was created in 1998 with a promise to find cures for neurodevelopmental disorders. Every day, our physicians and researchers come closer to fulfilling that promise. Their groundbreaking research on autism, Fragile X syndrome, chromosome 22q11.2 deletion syndrome, ADHD, and other brain disorders are helping children achieve their fullest potential. Please visit our website to find out more about current studies, upcoming events, and how you can help make a difference.